Hello all, welcome to my channel. In today's class on antibiotics, we'll be looking into two antibacterial antibiotics and one antiviral agent. For all the three, we'll be focusing mainly on their source, the structure of that antibiotic in brief, the spectrum and its mode of action. So starting with the first antibiotic, that is polymyxin. Now, polymyxin is an antibacterial agent, which means it is active against bacteria or it is killing or inhibiting the growth of bacteria. The antibiotic itself is isolated or it was first obtained from another species of bacterium, that is Bacillus polymyxa. Now, Bacillus polymyxa is a gram-positive rod-shaped bacteria and this is the source for the antibiotic polymyxin. Even though the antibiotic is active on bacteria, it is a bacteria itself which is producing this particular antibiotic. So as you can see, it is a gram-positive rod-shaped bacterium and this is how the colony of Bacillus polymyxa looks. Now the structure of antibiotic polymyxin it is a cyclic polypeptide. When we say it's a polypeptide, it means it is having several amino acids that are attached to it in a cyclic manner. That is why we call it as a cyclic polypeptide. This antibiotic is active only on the gram-negative bacteria, or it is active more on the gram-negative bacteria, because of which we classify it as a narrow spectrum antibiotic. So this antibiotic is acting mainly on the gram-negative pathogens because gram-negative pathogens are made up of an external layer of outer membrane. This layer is present outside their cell wall. Now this outer membrane is not there in the gram-positive bacteria and it is this outer membrane that the antibiotic goes and acts upon. That is why all the gram-negative bacteria which are having this outer membrane are more susceptible to polymyxin antibiotic in comparison to their gram-positive counterparts. Now what this antibiotic does is, it is acting on the lipid layer. As we all know, the lipid layer or phospholipid layer is mainly found in the plasma membrane or the cell membrane of an organism, and it is also found in the outer membrane of gram-negative bacteria. So those cells which are rich in phosphatidyl ethanol amine, which is a type of lipid. So if the cells are having more abundance or they are having more concentration of phosphatidyl ethanol amine, such cells are more susceptible to this antibiotic. The reason is polymyxin has a detergent-like action. So it binds to these phospholipids which are found in the outer membrane or in the plasma membrane of an organism. And as soon as it binds to the outer membrane or it binds to the plasma membrane, it leads to the damage of that membrane. It leads to pore formation, or there are channels being formed. Through these channels or through these pores, all the contents of the cell are getting leaked out. So there is leakage of the cell contents which leads to the cell death. That is the mode of action of polymyxin. So polymyxin binds to the phospholipids which are present in a cell, and after binding to the phospholipids, it leads to the formation of pores which in turn results in the leakage of contents of the cell. The next antibiotic that we will be looking into is an antiviral agent, that is acycloguanosine. Now in general, we see that it is very difficult to treat viral infections because viruses survive inside the host cell. So if I have to target a virus, I have to first target the host cell. I have to, in many cases, kill the host cell, and then only I can reach the virus or I can destroy the virus. So what it means to say is that in my quest to kill the virus, I have to have collateral damage of the host cell because my virus lives inside the host cell. That is one major problem with the development of chemotherapeutic agents that kill or that attack viruses. The other reason why viruses cannot be easily targeted is because they don't have much proteins or they don't have much particles that they produce. They are using the host machinery. Most of their enzymes are taken from the host. Their ATP is taken from the host. So if I try to target a particular enzyme, it will be the host enzyme that I will be killing, and it is not a viral enzyme. So the two reasons why it is difficult to develop an antiviral agent are: one, that the virus is living inside the host cell, and secondly, the virus is using the host machinery. So that makes the treatment not very effective. We do have treatment strategies for antiviral infections, but they are not very effective due to these two reasons. So these chemotherapeutic agents which are effective against viruses are of different types, like you have interferons, or they are RNA polymerase enzyme inhibitors, reverse transcriptase enzyme inhibitors, or a very popular choice of antiviral drugs are nucleoside analogs. And acycloguanosine is exactly one such nucleoside analog. So let us look into it in detail. The source of this antibiotic is a sponge. It's a sea sponge, that is Cryptotethia crypta. Now, it was in the 1970s that they identified particles, or they identified substances which were having this kind of a structure from the sponge. So, it was first isolated from this Cryptotethia, which is now known as Tectitethia crypta. But what was isolated from this sponge has been the basis for acycloguanosine or acyclovir. That is another name for acycloguanosine, that is acyclovir. So there were compounds that were isolated from the sponge which were having, or which were analogous in structure, or which were having a similar structure to that of nucleosides. That is how we came up with the idea of acycloguanosine. That is how acycloguanosine was synthesized. Now, it was initially discovered in the 1970s, and it is very important, or it was a big hit at that time, or it was a breakthrough, because it is selectively toxic to viruses. That is mainly because this particular acycloguanosine has to get phosphorylated by a viral enzyme. So the virus has to produce one enzyme that is called as thymidine kinase, and only if that enzyme is produced by the virus and that enzyme is phosphorylating this, it will act as a nucleoside analog, or it will do its work. So we look into what exactly its mode of action is, but the source is a sponge, that is Cryptotethia crypta. The structure, like I have been telling from the last 5-10 minutes, is that it is a nucleoside analog, which means the structure of this antibiotic is very similar to the nucleoside deoxyguanosine. What you see on the right over here is deoxyguanosine, which is made up of a deoxyribose sugar and the guanine base. This is called as a nucleoside. So this is the nucleoside deoxyguanosine, made up of guanine base and the deoxyribose sugar. This is what gets incorporated into the DNA of the viruses. On this side, as you can see, we have the structure of the antibiotic or the antiviral agent acycloguanosine. As you can see here, the guanine part of it has been exactly replicated in this antibiotic, and we also have the deoxyribose sugar, which is not present here, but we have a similar OH group at the end as we have in the case of deoxyribose sugar. So most of the structure of this antiviral agent is similar to that of the deoxyguanosine nucleoside. Now that exactly is its mode of action. So when it is having structural similarity to a nucleoside, this is what helps in us using this as an antibiotic. We'll look into it a little later. Now the spectrum of this antibiotic is, it's a broad spectrum antibiotic because it can be used for the treatment of viral infections. Wherever there is DNA involved, we can use this particular antibiotic or antiviral agent. And mainly acycloguanosine is used for the treatment of HSV infections, that is herpes simplex virus infections, and varicella zoster infections. These are the viruses it is mainly used for, but it can be used for any DNA virus. Now the mode of action, coming back to the mode of action, I told you that it is having structural similarity to that of a nucleoside deoxyguanosine, which means if this is administered to a virus, this gets phosphorylated, and when it gets phosphorylated, it forms a false nucleotide. So as you can see over here, this is guanosine this is deoxyriboguanosine, which gets phosphorylated, whereas now here it is our acyclovir or acycloguanosine which gets phosphorylated. Now when this gets phosphorylated, it forms a false nucleotide, which means it is unable to do polymerization with the other nucleotides of DNA. So even though the nucleotide is ready, we are not having DNA polymerization occurring over there because it is unable to get polymerized. It is unable to form a new DNA for the virus. So this is its main mode of action, wherein it is resembling it is similar in structure to that of the original nucleoside, and when this gets phosphorylated, when this antibiotic or antiviral agent gets phosphorylated, it is stopping the DNA polymerization, or it is hindering the DNA polymerization, because the phosphorylation, till the phosphorylation occurs, it's okay, but after that it is unable to take part in the DNA synthesis. So this was about the antiviral agent, that is acycloguanosine. Coming to the last antibiotic for today, that is nalidixic acid. Now, nalidixic acid is also an antiviral agent, an antibacterial agent. But why it has been spoken about separately is because it is a synthetic chemotherapeutic agent, which means it has never been isolated from an organism. It is being completely synthesized in the lab. Hence, it is called as a synthetic chemotherapeutic agent. Now, the structure of this antibiotic is, it is a quinolone compound, and it was first used in the 1960s. So, it was synthesized in the lab and it has been used ever since. The spectrum of this antibiotic is, it is a broad spectrum antibiotic because it is able to affect gram-negative bacteria, many gram-negative bacteria, and even a few gram-positive bacteria. In fact, the gram-positive bacteria it affects are extreme pathogens. For example, Bacillus anthracis, which causes anthrax. So, nalidixic acid is able to act on such gram-positive pathogens and even many gram-negative bacteria such as E. coli, Proteus, Enterobacter, Shigella, Klebsiella, many gram-negative bacteria, as well as some very important critical gram-positive bacteria such as Bacillus anthracis. Hence, the spectrum of this antibiotic is a broad spectrum. The mode of action is, this antibiotic too inhibits the DNA replication, but the part of DNA replication or repair that it inhibits is this antibiotic, that is nalidixic acid, binds to the DNA gyrase enzyme. Now, DNA gyrase is an enzyme which is present in the prokaryotes, which is helping in introducing negative twists in the DNA or negative supercoiling in the DNA. Now, if the DNA gyrase enzyme is not present, what happens is that there is no negative supercoiling that is occurring, and that can affect the DNA replication or even the DNA repair process. So, DNA gyrase is the enzyme that helps. You can see over here in this diagram, this is the DNA gyrase enzyme. It binds to the DNA and helps to introduce negative supercoiling or negative twist, so that when the strands of DNA are separated for the DNA polymerization or DNA synthesis, at that time, to maintain the structure of the DNA, this DNA gyrase enzyme is binding to it and introducing negative supercoiling so that the structure can be maintained. Now, when the DNA gyrase itself is not functioning due to binding of the antibiotic, automatically it inhibits the DNA replication and repair mechanisms. So, this is what we have done in today's class, the three different antibiotics, that is polymyxin, acycloguanosine or acyclovir, and nalidixic acid. As you can see, there is no source for nalidixic acid because it is a synthetic chemotherapeutic agent. So, these are the different antibiotics that we have done today. Two antibacterial agents. One was a natural antibiotic which was produced from a bacteria. The other one is a synthetic chemotherapeutic agent. And we have also spoken about a very important antiviral agent. See you all in the next class. Thank you.